Well, it's time to talk about DNA. It's kind of a central topic to biology. DNA is used, if you remember, to do the best classification we think we can on life on Earth. And it's DNA that manufactures proteins that make elephants and giraffes and whales and at one time dinosaurs and oak trees and bushes and ferns and grasses, etc. So we got to talk now about like what is DNA and how does it work. So DNA is short for deoxyribonucleic acid. It's the largest molecule found within typical cells. It has a program in it. It's sort of like code that you would write to do a computer program in a way, but it's biological code. We call it genetic code. And what this genetic code does is it allows a cell to function. The genetic code is written basically in the sequence of nucleotides that are in the DNA. So what a chromosome is basically a bunch of nucleotides strung together, usually at least millions, if not billions, strung together in sequence. And it's the specific sequence of nucleotides that specifies what kind of proteins that the DNA can make. Basically, that genetic code is passed from one generation to the next through reproduction. Biology didn't stumble on DNA until about 1871. Friedrich Miescher discovered it, but he thought it was too simple to be a hereditary substance. And by 1901, chromosomes were observed separating during sperm formation. And it was thought that chromosomes were probably responsible for heredity, but chromosomes by mass are about 50% DNA and 50% protein. And at the turn of the 20th century, the protein was recognized as more complicated than the DNA. So it was believed at that time that the DNA was there just for structural purposes and the proteins actually stored the hereditary information. So we fast forward a little bit to 1928. A British health scientist named Fred Griffith, working for the British Health Service on vaccines with strep pneumonia, was trying to come up with a pneumonia vaccine. Pneumonia was a leading killer at the turn of the 20th century. And it still is a formidable opponent today. People still die of pneumonia today. So Fred was trying to come up with a vaccine, and basically he was injecting all kinds of pneumonia cocktails into mice to see if he could come up with a vaccine and then keep the mice dying from actual pneumonia. Fred is credited with discovering a non-virulent strain of pneumonia that didn't actually kill mice. He was actually excited about that at first because he thought that maybe those non-virulent bacteria would serve as a vaccine. But as it turned out, the non-virulent bacteria didn't work out as a vaccine. But he was persistent. He tried different combinations of live non-virulent bacteria and dead virulent bacteria to see what would happen if he could come up with a vaccine. So when you do vaccine experiments to try to develop ways to treat diseases, you have to, usually what you do is you kill somehow the virulent microbes that you have, and then you inject them into a mouse, and you hope that the mouse develops an immunity to whatever the dead stuff was, so that when you inject living bacteria or parasites into the mouse's immune system will recognize those and kill them before they can kill the mouse. So here's a couple of the experiments that Fred was doing. He injected live smooth bacteria, these are lethal, into mice. The mice died. When you cultured the blood of the mice, there were live smooth bacteria in the blood of the mice. He then tried live rough, hoping that these bacteria, which are Pneumonia bacteria. These are pneumonia bacteria. They appear rough on the surface under the microscope. He was hoping that when you injected those into mice, the mice not only would live, but you would be able to culture live rough from the blood of the mice. And then if you injected the smooth strain of the bacteria into these vaccinated mice, they would survive. They did not. But in one of his peculiar experiments, he tried a combination of dead smooth, that's these, they're gray, and live rough. And he hoped that this combination would produce a vaccine. To his surprise, when he injected this cocktail into mice, the mice died. When he cultured the blood of the mice, there were live, smooth bacteria in the blood of the mice. Now remember, he didn't inject any live, smooth bacteria. And even in 1928, they didn't believe in spontaneous generation. These bacteria could not spontaneously, become alive again. So Fred's hypothesis was that these gray bacteria, transformed somehow these live roughs into killers, into smooth variety. And so Fred began to work on determining the identity of the transforming principle, but Fred died in one of the German bombing runs of London in, I believe, it was World War II. And so Fred never got to figure out the answer to what he had stumbled on. So another team of scientists, Oswald Avery, Macklin McCarty, and Colin MacLeod, in 1944, separated live, smooth bacteria into 4 components that make them up: the DNA component, the protein component, the lipid component, and the carbohydrate component. When they mixed the carb component with live rough and shot them into mice, the mice lived and they had no bacteria in their bloodstream. That's because the immune system of the mouse killed the rough bacteria. When they shot lipid and live rough into mice, again, no bacterial transformation occurred. The mice lived, and there were no bacteria in the bloodstream of the mice. When they shot protein, which now is a surprise, with the live rough, the mice lived, and the mice had no, culturable bacteria in their bloodstream. When they shot DNA from these dead virulent bacteria, along with live rough bacteria, the mice died, and they died of this strain of bacteria. We call this bacterial transformation, where these bacteria pick up pieces of DNA and become transformed and turn into a different strain. We use bacterial transformation in the lab today for all kinds of things, but this is the very first discovery of how it worked back in the early part of the 20th century. So this was amazing for a couple of reasons. One, that you could transform bacteria from one strain to another. Two, that it was only the DNA fraction that caused the transformation and not protein. So this seemed to lay to rest the idea that protein was hereditary, but a lot of biologists still bought into the concept, and they just would not believe that DNA was complicated enough to store hereditary information. Proteins are made up of 20 different amino acids, but DNA is only made of 4 different nucleotides, hence the problem. So, Alfred Hershey and Martha Chase in the early 1950s did a classic experiment with viruses. And what they did was they used radioisotopes, these are radioactive atoms of phosphorus and sulfur, in the viruses to see what the viruses would inject into bacteria when the viruses attack bacteria. Now, we talked about viruses in a previous unit, and I mentioned that when viruses attach to cells, they inject something into the cells. And Hershey and Chase wanted to know what the something was. And based on Avery, MacLeod, and McCarty's isolation of DNA as the transforming principle here, they figured it was DNA. So what they did was they allowed viruses to attack bacteria, and when the viruses did, they only found phosphorus inside the bacteria but not sulfur. Now, as it turns out, the viruses, this is a typical bacteriophage or bacteriovirus. DNA contains, if you remember from the molecular biology unit, carbon, hydrogen, oxygen, nitrogen, and phosphorus. So the radioactive phosphorus is in this red part. The protein, okay, which goes all the way around. It's the tail piece here, which is the injection assembly for the nucleic acid, and this protective shell, that's got some sulfur in it. So the radioactive sulfur is in this bluish part, and the radioactive phosphorus is in the red part. When they let the viruses attack bacteria, they only found radioactive phosphorus inside the bacteria. What that indicated was that viruses were injecting only their nucleic acid into the bacteria for the assembly of new viruses. So the Hershey-Chase experiment showed that at least for these kinds of viruses, the hereditary material was definitely DNA and not protein. And that pretty much began the final page of protein as a hereditary material. Most biologists switched camps and believed then that it was DNA that was the hereditary chemical and not protein. Now, some other scientists had done some things that seemed to describe the quality of DNA in cells. Erwin Chargaff in the late 1940s, studying adenine, thymine, guanine, and cytosine, which were the 4 nitrogen bases in the 4 different nucleotides of DNA. He found that always the ratio of, it says, adenine to guanine for a particular species was absolutely constant. Furthermore, he always found that the amount of cytosine in the cell equaled the amount of guanine in the cell, and the amount of adenine in the cell always equaled the amount of thymine in the cell. Again, but not only that, he found that for a particular species, no matter when or where you tested it, the ratio of A to G for the species was always the same. So he was stumbling on a concept that somehow written in the nucleic acid was some kind of a message that determined what the species was. Now comes Rosalind Franklin. Rosalind Franklin in 1951 was doing X-ray crystallography. She was the only person in the world doing this. Rosalind probably died of the X-ray exposure. She died in the late 1950s from breast cancer. She knew that DNA was composed of adenine, guanine, cytosine, thymine, deoxyribose, and phosphate. Now, she was trying to figure out how those 6 components made up the structure of DNA. Everybody knew that the first person who discovered this structure was going to be absolutely famous. Now, Rosalind was the only person in the world that was actually trying to figure this out. And she was trying to build a model of DNA based on her crystallographs. And what a crystallograph is you very carefully create a crystal of a chemical in a very specific way, and then you shoot X-rays through it. And it's sort of similar to looking at an X-ray of a bone. You can tell, whether was the bone broken and qualities of the bone from the X-ray. Rosalind was trying to determine the position of adenine, guanine, cytosine, thymine, deoxyribose, and phosphate in the DNA from her crystallographs. Unfortunately, Maurice Wilkins, one of Rosalind's lab assistants, passed along without Rosalind's knowledge some really high-res photographs or crystallographs of her DNA to James Watson and Francis Crick. And Watson and Crick were really good biochemists, although they were brand new. They were just out of graduate school. And using Rosalind's, stolen DNA crystallographs, they developed the structure of DNA. And they hastily published a paper in 1953, and they got the Nobel Prize in 1962. They never actually admitted that they used Rosalind's high-res photographs. They implied to the world that they had just figured it out. It's one of the greatest lies in the history of 20th century science. Today, James Watson won't even talk about it, even though he is now kind of vilified by a lot of people in biology because he took advantage of Rosalind and didn't even want to admit until many, many years later that this had happened. And he actually treated her rather badly. By today's standards, he'd be fired from the lab for doing what he did. So what Watson and Crick proposed, and we should call it the Franklin model, and there's a movement to do that now. What the Franklin model was presented as is the reason the same amount of adenine and thymine are present in the cell, and cytosine was always the same as guanine in the cell, is it was caused by a phenomenon that we know of as base pairing. So it turned out that the way DNA was structured was, this is a simple model of DNA that James Watson and Francis Crick had put together. Here's a deoxyribose, here's a guanine. Across from the guanine is a cytosine, and here's a deoxyribose attached to the cytosine, and there's a phosphate. Each nucleotide is composed of three components: a phosphate, a deoxyribose, and a nitrogen base. Phosphate, deoxyribose, cytosine. Phosphate, deoxyribose, adenine. Phosphate, deoxyribose, guanine. Phosphate, deoxyribose, thymine. On the opposite side, the phosphates face the opposite direction. On the opposite side, we have phosphate, deoxyribose, adenine. Phosphate, deoxyribose, cytosine. Phosphate, deoxyribose, thymine. And then lastly, we have phosphate, deoxyribose, guanine. So you can see the reason Erwin Chargaff stumbled on this is this phenomenon where the guanine is always equal to the cytosine and the adenine is always equal to the thymine is because of this base pairing. The adenine cannot bond to guanine. It cannot bind to other adenine. It cannot bind to cytosine. It can only bind to thymine. So they're always found in a 1-to-1 ratio. For every one thymine, there will be one adenine. For every one cytosine, there will be one guanine. This is a three-dimensional arrangement of the atoms. It's actually a twisted, kind of like a spiral staircase, is the way it's constructed. So Watson and Crick actually did successfully develop the model of DNA. This is a picture of Rosalind here. She didn't get the Nobel Prize. She died before the Nobel Prize was awarded for this research. There's one of her low-resolution crystallographs. And this is right here, I believe, this is Macklin McCarty of Oswald Avery and McCarty that had discovered that it was DNA that transformed the bacteria. This is James Watson, and that is Francis Crick. These are the two that actually behaved rather badly using research from another scientist. Now, that concludes our discussion of the structure of DNA, except for one last topic I guess I should mention. You see where it says in this model 5' end, and down here it says 5' end? As it turns out, DNA is actually a double helix. This, on this side here, this is a rope. It's a rope. See, here's the rope. And then there's these nitrogen bases sticking out the side of the rope, like little hairs sticking out. On the other side, we have another rope here, and the nitrogen bases are sticking out. And these nitrogen bases stick to these nitrogen bases, so two ropes essentially are attached to each other. It turns out that the two ropes are anti-parallel to each other. The rope on this side, the 3' here, is referring to the number of the carbon. This is carbon 1, carbon 2, carbon 3. That's carbon number 3 of the deoxyribose. Over out on this end, we have carbon number 1, carbon number 2, carbon number 3, carbon number 4. That's carbon number 5 there. The number 5 carbon of this deoxyribose is facing up. The number 3' of this carbon the number 3 carbon of this deoxyribose is facing down. On the opposite side, the number 5 carbon of the last deoxyribose, it's facing down. And at the top up here, this number 3 carbon of this deoxyribose is facing up. The two sides, which are ropes of nucleotides, are opposite to each other, and they face oppositely, and that offers some peculiar DNA behavior that we're going to talk about a little bit later in this unit.